and what they are, what the identifiers are. So just starting off, what is this? What are we looking at?
My bad.
I think I heard it. What? Pituitary, good. And so this is pituitary kind of in a slight panoramic. You don't see the whole endocrine gland, but you can see that there's two very different spots, right? This upward area, the upper portion of the slide, and the lower portion of the slide look very different. So what is going on here? Is this all pituitary, or is just the top part pituitary, just the bottom part? Any thoughts? Okay, yeah, both halves, a little divider, good. Okay, so what's the divider? What's its name other than the little divider? That won't count on the practical. So, yeah, I think I heard it. Pars intermedius. Yeah, it's because it's intermediate. It's intermediate or between these two portions. So what are these other two portions then? If this is the intermediate area, pars distalis. Good. And pars distalis is where? Is this pars distalis or is this bottom portion pars distalis? Top. Good. So the top is pars distalis or the anterior pituitary or the adenohypophysis. If you're in my sections, all of those names are acceptable in the practical. Any of those terms. In terms of the pituitary in general, pituitary gland hypophysis is acceptable. If you're in my sections. How do we know, though, that this is pars distalis? What tells you that this is pars distalis and not this bottom portion? Granules, okay, yeah, there's some granules in here too, but there's a lot more granules here. Why are there more granules in pars distalis? So pars distalis, we've got lots of cells, right? And then this bottom portion, which must, by process of elimination, be, this is pars distalis, this is pars intermediate. What is this third? What's the third portion of the pituitary gland that we learned about in the lab? Nervosa, good. So nervosa, meaning that it's what type of tissue? Nervous tissue, right? Nervosa, nervous tissue. And so what's the big difference between pars nervosa or the neurohypophysis or the posterior pituitary gland and the anterior pars distalis adenohypophysis? What's the big, aside from that, there are two different parts of the pituitary.
The other one's glandular?
Which one is the other one?
The one that's not nervous?
The one that's not nervous is endocrine cells. So not really glandular per se, but it's mainly cells. This is all. It's got this stippled dotted appearance because it's a lot of cells. And down here we don't have as many cells. It's nervous tissue. We have lots of axons specifically. Yeah. So the axons of neurons. And remember, pars nervosa is an extension of the hypothalamus, and so you have the cell bodies in the hypothalamus with their axons extending down into the posterior pituitary gland. So you have lots of axons in here, and then those neuroglia, those supporting cells, are primarily the only cell bodies that are in here. And so you have the hormones being produced in the cell bodies up in the hypothalamus and then being sent down the axon into pars nervosa. Cell-wise, in the anterior pituitary, what cells do we have? What are our big cell populations in the anterior pituitary of the adenohypophysis, pars distalis? I told you that they're named based on color. Would that be helpful?
Like
basophils, good. And so basophils are usually bluish, purple kind of colored. And then there's also acetophils, good. And so those two collectively, they're colored, so they make up the chromophils, the cells that like color, that absorb stain because they have the granules for the different hormones they're secreting in them. And then we've got a third population that doesn't like color. So if they don't like color, instead of being a chromophil, they should be a chromophobes. Okay, so they also have the chromophobes, which should be clearish, right? So the acidophils are red, basophils blue. Chromophobes, they don't like color, so they don't have color, and there's no endocrine hormone granules that are in there to pick up the stain, which is why they're not colored. Good. So then, zooming in a little bit. So now we're just looking at one segment of the pituitary. If you had to take a guess, what segment this is, what would you? What part? Pars distalis, yeah, or the adeno hypophysis. Anterior pituitary, you can see all the cells, right? So it's got to be anterior pituitary because there's a cell population here. You can see the granules in the cytoplasm. They've got clear nuclei over the place, and then you can see that there's differences in the cells. There's a couple up here that have arrows going to them that are reddish, and then down here they've got some blue dots in them. The whole cell body isn't blue, but there's blue dots, and then down here these two arrows are going to cells whose cells bodies are clear. So those would be, again, our acidophils, basophils, and chromophobes. So chromophobes being these clear cells, the basophils being the ones that have the blue dots, and the acidophils being the ones that are reddish, or in this case they're kind of more like magenta, but somewhat red. In the anterior pituitary, what types of hormones are produced? There's 7 altogether in the anterior pituitary. Shout them out. ACTH, which stands for... Okay, good. Yeah. So if you're in my sections, there's no acronyms on the practical. You need to know the ACTH won't count. You need to know the whole thing. What else is in the anterior pituitary? Okay. Melanocyte stimulating hormone. Human growth hormone, HGH, good. Yep. Follicle stimulating hormone. What one, Sam? Luteinizing hormone. Good. What is that? That's 5 or 6, I think. Anybody say somatotropin? What do you mean when you say about it? Like it's pathway or? Okay. Okay. Alright. Yeah, good. So there's your hormone secretions for anterior pituitary. Jumping to the other side of the pituitary. Now we have a close-up of what part of the pituitary? Pars nervosa. You can see the nervous fibers, right? And then most of these cell bodies that are in here are going to be the neuroglia, so those neuron supporting cells are taking care of maintaining, directing the axons that are in this tissue. Secretion-wise, what hormones do you find in the posterior pituitary, the neurohypophysis? Oxytocin and ADH. So ADH stands for, well, it's also vasopressin. That's another name for it. So vasopressin, and if you call it antidiuretic hormone. Good. All right. Good. All right. Now we're switching. What endocrine gland is this? I think I heard it. Thyroid. And how do you know it's thyroid? You can see follicles. Good. And colloid inside of the follicles. Around the follicle, what should these cells be? The ones that are right on the perimeter of the follicle? Follicular cells. Good. They're at the they're a part of the follicle. They're follicular cells. In between these 3 follicles, there's another cell population. What cells should that be? C cells, yeah, or also parafollicular cells. Either is acceptable if you're in my section on the practical, C cells or parafollicular cells. C cells are called C cells because they're secreting what? Calcitonin. Good. Huh? And then what types of hormone products do you expect to see from follicular cells? T3 and T4. What do T3 and T4 stand for? Triiodothyroxine, right?
Do we have to know that?
I think those will probably be the only exception that I would make in terms of acronyms, because you're all going to spell them super crazy anyway. The other ones, if you're in my section, everything except for T3 and T4, you would need to know what that acronym stands for. Okay. Let's see. So then, moving along, we can see what out here. We just looked at it. Yeah, so the follicles of the thyroid again, and then there's this lump, this mass. What is this mass? It's not cancer. Parathyroid, good. Alright, so we've got thyroid all around here, and then if you go to the posterior side of the thyroid gland, there's these little lobes, these little kind of dots on the back of it. Usually there's 4 of them, and that's the parathyroid. You can see a major difference in the cell population, right? There's no colloid in the parathyroid. The cells are kind of clustered in bundles. You don't have this arrangement structure with these huge lumens that cells are around. What secretion-wise do you expect to see from the parathyroid? Parathyroid hormone. Good. Okay. Now it's kind of a little more easy to remember. It's the parathyroid and has a parathyroid hormone. And then again, just a close-up of parathyroid, kind of, really hit home the fact that this is very different than the thyroid. No colloid, right? Cells are more closely arranged. You don't have these massive lumens. They're not that same slender type of arrangement. So very, very different tissue between those 2 endocrine glands, even though they're right next to each other. Right. Moving along, kind of another sort of panoramic. What endocrine gland is this? You can see there's lots of different layers. Adrenal gland, good. We work through those layers starting from the top of the slide to the bottom. What are they? So this top thin one that's got kind of clustered or ball-shaped, grouping of cells. This is zona glomerulosa. The cells are arranged in glomeruli. Good. And then moving down, you can't see it too well in this particular slide, but then nice parallel columns of cells. That should be zona fasciculata. Good. And then continuing down further, another thin kind of layer. Those columns sort of drop out, and instead you have these kind of interdigitating or woven stacks, ropes, so reticularis, good. And then moving down to the very bottom of this, medulla, good. And so, what part is the outer region of this gland? Is medulla the outer region, or is glomerulosa the outer region? Glomerulosa, good. And the medulla is in the middle, right? Medulla, middle, and then glomerulosa, fasciculata, and reticularis make up what region? The medulla is the middle. The cortex. Okay, good. Good. Let's see. Okay, so then zooming in a little closer, you can see here you've got, again, those clusters, so this must be zona glomerulosa, and then just beneath that is going to be fasciculata. You can kind of see the columns better with this. The cells are arranged in little ropes or columns that are going, perpendicular to the outer portion of the cortex. Secretion-wise, generally, not anything super specific, but generally, what does glomerulosa secrete? So, mineralocorticoids, good, and then, so fasciculata is your glucocorticoids. And then, moving in a little further, now you can see, so what are we looking at now? We're continuing to move in. Now you can really see that nice parallel kind of structure, these columns. So this is fasciculata still, and going in deeper. Now, what layer do we have? Kind of going back to this sort of, woven is the wrong word for it. So this is medulla. And I don't think I don't have a slide of just reticularis. Okay. Okay. Well, so reticularis would be out here somewhere, if I remember how this slide is oriented. But yeah, medulla now. Going back to reticularis, what is reticularis responsible for hormone-wise? Androgens. Good. And then in medulla, you have epinephrine and norepinephrine. Good. Let's see. Need to move along. This one doesn't have the best distinction in the world, but if you look, you can kind of see these light little isolated patches throughout the whole tissue. And so there's the darker areas and then the lighter areas, and they're like these little islands of lighter staining cells. So what endocrine gland do we learn about that has kind of a mix of endocrine with other stuff? Pancreas. Okay, good. So this is the pancreas. The endocrine portion is going to be lighter. Okay, yeah, and specifically it would be called the it's Langerhans. Good. You can see them here. They kind of look like baseball gloves a little bit. And then so all this dark then is the exocrine portion of the pancreas. And then here's a close-up of those islets of Langerhans. You can see you've got these ropes that are all weaving into this glomerular shape, and there's red blood cells through here, right? You can see vasculature. Why? If we go through all of these endocrine glands as we're going through, we're always going to see vasculature. Why is there always vasculature present in these endocrine glands? Because they're making hormones, and so why do you need vasculature with hormones? Yeah, so you need that transport system that as the cells secrete it, it goes into the blood to be delivered to distant targets, right? Endocrine signaling is to a distant target. All right. Cell population wise, if you look at the islet of Langerhans stain, you can see there's different colors going on here. There's red, blue, green. So these colors represent different cells that have different products that are picking up a different colored stain. We focused on just 3 different cell populations in the islet of Langerhans. What were they and what did they secrete? So alpha cells, and they were responsible for glucagon, good, and then beta cells, which were responsible for, good, and then the last one that we learned about, yep, and that was somatostatin, good, not to confuse with somatotropin, somatostatin, good. Alright, good.
What's the function of somatostatin?
Somatostatin is body. I don't remember. I don't remember. So obviously you don't need to worry about it either. I don't remember that kind of good question though, Sam. Thank you. Sorry. It's involved with both. A growth hormone inhibitor?
Yeah.
Okay.
Yeah. Which I guess somatotropin would be a growth hormone stimulator then?
Somatotropin should be involved with HGH. Yeah, but again, I don't know the pathway, or I don't remember the pathway anymore.
I only have one sentence, but it's from my
Okay.
actually my
Alright, so moving on, another endocrine gland. We've got these nice triangular shaped lobes, and you can see a distinction with this light medulla, dark cortex. What endocrine gland is this? It's got these triangular lobes with a light medulla, dark cortex. Thymus. Good. And if we look the lobes in this picture, nice and triangular. It's basically just the the cells of the thymus that are present. If we skip, the triangle kind of looks a little more disjointed. You can see there's invasion of connective tissue. If we looked closer, we'd see adipocytes with invasion of fat, adipose tissue. So what's the difference between this segment of thymus and this segment of thymus?
Just
Age, yeah, okay, good. And so which is older then?
the second one.
The second one. Alright, good. So this one that's got the connective tissue fatty invasion is going to be adult thymus, and the more robust looking thymus is going to be infant, juvenile thymus. And in this adult one, is it still functioning, or is it like it's atrophy, and that's why you have the connective tissue and fat in there? Is it still working thymus?
if you've got an immune system, right?
If you have an immune system, yeah. So it's the thymus is super active when you're young, when you're first born, building up your immunocompetency as you encounter all these different things in your environment with a more like a blank slate immune system. Then once that's more established, it starts to involute, right? You get the connective tissue fat invasion, and it doesn't become totally inactive, but it's no longer quite as busy building up immunocompetency, because by that point in time, you've become immunocompetent, and the amount of novel things your body is coming into contact with that it's trying to defend itself against is not at the same rate as you just, you've experienced your environment for a while. This is pre-pubescent. This should be a pre-pubescent change. The super hyperactive activity is in the, I don't know the exact cutoff, but early years of life, like early years of life, and that's part of the reason why it's so important to drink or to breastfeed when when you have a child, because a lot of that immunocompetency is transferred through breast milk. Or why it's not so important or good to breastfeed, because the immunocompetency is transported that way, that the mother's environment then puts in antibodies that the infant can take in through breast milk. It shouldn't be a puberty thing that involution starts. It should be earlier, because by then you should have a lot of your immunocompetency. Okay. Does that answer your question, or? Yeah. Okay. All right. So now we've out of endocrine, moving into the neuroanatomy stuff. This is kind of, again, more of a panoramic. What are we looking at? The section of brain. You can see there's a couple different layers. We only really looked at two different regions of the brain, right? We've got two options, right? So they would be cerebrum or cerebellum. Yeah, so it's either cerebrum or it's cerebellum. Cerebellum is probably the more easier to, well, there's only two options, but more easy to quickly identify. So what is it about the cerebellum that is super easy right away that you see and you know it's cerebellum? Okay, squiggly. Maybe, yeah. Okay, so if you're far enough away, you will see sulcuses and gyruses earlier on. Is that what you mean by squiggly? Okay. But we'll go closer. Cell population-wise, what's one of the big defining ones for the cerebellum? It's named after a researcher who we've already looked at. Something else that he is named after him. Golgi's one of them. Yeah, so there's Golgi stuff in there. But the big one is we learned about in the heart. We looked at cardiac muscle. We looked at cardiac muscle. We looked at specialized cardiac muscle cells that were called Purkinje fibers, and then so in the cerebellum you have Purkinje cells. Purkinje cells in the cerebellum, which are there Purkinje cells? They're real big fat cells, and they're right on the border of dark and light area in the cerebellum. Do you see any sort of cells like that? A whole bunch of big round fat cells in a line at this border between a light region and a dark region in this tissue? There's some dark here and some light here, but I don't really see super fat cell bodies, right? So this probably isn't cerebellum, which means it must be cerebrum. And in the cerebrum, there was another cell population. They're not round. They have another shape that was very distinct. They're named for their shape. What was that cell population in the cerebrum? Pyramidal cells, right. So if they're pyramidal cells, they're named for their shape. That shape must be a pyramid or triangle. So they've got large pyramidal cells, small pyramidal cells. And if at this level, you can kind of see where cell bodies are, but you can't exactly tell that they're triangular and that they're pyramidal cells. So if we go closer, you can see here's pyramidal cells, right? And they're in these nice triangles. The rest of the nuclei in here, if pyramidal cells are neurons, the other cell bodies that are small in here should be, what do you usually always have with neurons to take care of them and support them and direct them? Neuroglia, right? So these other cell bodies should be neuroglia, and then here's our pyramidal cells. And in the cerebrum, you have layers of pyramidal cells, and they're going to change in size. You'll have smaller pyramidal and then larger pyramidal as you head towards the medulla, away from the cortex. The layers we talked about briefly, but for the cerebrum, you don't need to be quite as concerned with them. However, if we switch, and then look at now cerebellum, the layers are much more distinctive, right? There's this white area where you can clearly see it's opaque, and there's just some cell bodies dotted throughout it. And then it's surrounded by this much darker layer that's very, very full of cell bodies. Basically all of this is, cell bodies, closely, densely packed throughout the tissue in this region. And so these two layers are the, what two layers? So the molecular layer, right? And then the granular layer. And right at that border between molecular and granular, you can see these large, round, fat cells just in a line right on that border between those two layers, and those are going to be the Purkinje cells. And then out here, you've got your stellate cells and your basket cells in the molecular layer, and in the granular layer, this darker, densely packed area, the granular cells and the Golgi cells. I think. Do you talk about the nervous innervation pathways for the cerebellum in the lecture with Dr. Merritt? No? Okay. It's very complicated and very confusing. But if you don't study it in lecture, if you do study in lecture before we have our practical, don't worry about it for lab. I'm not concerned. Won't be asking questions about the pathway. Just the layers and kind of the cells that are present, and especially the Purkinje cells. The molecular layer is this lighter opaque region, and that's where you have stellate cells and basket cells. The stellate cells are kind of star-shaped a little bit. They're also usually smaller, and then basket cells, you can identify them by their axons kind of for a basket around the Purkinje cells in this border area. Their axons will come down and they'll kind of dip under like a basket around the Purkinje cell. I don't think you can really see that very well in any of the slides that we have, because I don't think any of them are stained for, and the one that I pulled for today, I don't think is stained for axons, so you probably won't see that too well, but the Purkinje cells you can definitely pick out, because they're these large, fat, round cells, and they're right on the border of dark and light, and they're usually kind of in a line, just all along here. Moving out of the brain, what is this a cross-section of? Spinal cord. Good. Everybody. Okay, so spinal cord. What were some important structures and important regions with the spinal cord? Gray matter and white matter. Good. Now, where is gray matter? Where is white matter in the spinal cord? Gray is inside, and in this case, it is the light-colored portion. Gray is this H here. And so then by process of elimination, the white must be outside. Yeah, all this outer area is going to be the white matter. So this is kind of a counterintuitive image. But what is it that makes white matter white matter? Why is white matter called white matter? Yeah, it has myelination, which gives it usually a whitish kind of look, depending on how you stain, or what type of stain you're using. In this case, obviously, it doesn't look particularly white, but this is going to be the white, and it's named that because of the myelination. And then in the gray matter, you could potentially still have myelination, but what is the big feature of gray matter that kind of helps distinguish it from white matter? Yeah, and so if you have rough endoplasmic reticulum, you must have what part of a cell? If myelin is going to be wrapped around an axon, where would you find rough endoplasmic reticulum? In a cell body. Okay, yeah, so cell bodies are going to be in this gray matter, and you really won't see many cell bodies, especially neuron cell bodies, in the white matter. It'll basically just be axons. And then supporting glial cells, obviously you have to have there. If it's myelinated, you have to have. The spinal cord is part of what part of the nervous system? Central nervous system. And so if you're myelinating an axon in the central nervous system, what neuroglial cell is doing that? Oligodendrocyte. Good. And if we moved outside, if we went out, through this structure, which is what part of the gray matter is this? Dorsal horn. Good. It's the dorsal horn because it's going all the way out to the periphery, right? So if we go all the way out through the dorsal horn and go out one of the posterior rami out of the spinal cord, out through the meningeal layer, you're then no longer in central nervous system. You're in peripheral nervous system, and in there, if the axon was stone myelinated, what neuroglial cell would be doing that? Schwann cells. Good. So Schwann cells, peripheral nervous system, oligodendrocytes, central nervous system. And for the EMs that we have out, if you look at a Schwann cell, how do you know it's a Schwann cell versus an oligodendrocyte? If you're just looking at the cells itself and you don't know where in the nervous system you are, what's usually your big indicator? Yes, so a Schwann cell is only wrapping one axon, whereas the oligodendrocyte will kind of centralize and extend out to a few axons and wrap them. So Schwann cells 1-to-1, oligodendrocytes 1 cell to multiple axons. And that's so aside from location, that's a structural difference in terms of neuroglial cell myelinating an axon, and that's an important distinction to make. It's when it's myelinating an axon that there's that difference between Schwann cells and oligodendrocytes. If these are the dorsal horns, what must these more rounded portions be? Ventral horns, good, and then laterally, we see these little triangles, those would be the lateral horns, okay, good. Where do you see lateral horns? They only occur in one segment of the spine. Thoracic, right, okay, good. Within the white matter, you're going to have ascending and descending tracts in terms of information. Where should an ascending tract be headed to, and what type of information should it be taking? Okay, sensory to the brain, good. So ascending, sensory to the brain, right? That makes sense. If it's going up the spinal cord, that means it's heading towards the brain. You want to take sensory information to the brain, right? And it would also be afferent, like an A, yeah. Afferent, ascending, afferent, okay, good. And then if it was descending, it would be going towards peripherally somewhere, good. Okay, and in terms of information, it's going to be motor, good. And then that would also be, as opposed to afferent, it would also be efferent with an E. Okay, good. Good. Now this is a close-up of spinal cord. So we're still in spinal cord, and you've got two different type of regions for the spinal cord. We just talked about them. So on the left here, this should be what type of matter? Gray matter. Good. Now you're closer. You can see the cell bodies, right? So here's your neuron cell bodies. And if we switch to the right-hand side, this is white matter, myelinated axons, and a lack of cell bodies. Good. Okay. Right. So out of neuroanatomy and into special senses. What special sense organ is this? Eye. Good. What are some big structures that stand out right away looking at this nice panoramic of the eye? The lens. Yeah, right. This big pink thing. Good. And so anterior to the lens, another kind of arch, whitish. What should that be? The cornea, good. And if we follow the cornea laterally and posteriorly, what is it continuous with in terms of the tunics of the eye? Sclera, good. And then going deep to the cornea, you've got these kind of wingy things on either side of the lens. Nope, too deep. That's too deep. These are kind of the middle tunic. So anteriorly, the middle tunic determines what color your eyes are. Iris, good. So here's your iris, and then that's going to be continuous laterally and posteriorly with the vasculature area of the eye, the vasculature tunic. So the choroid, right? So choroid is deep to sclera, and then in terms of function. Well, no. So continuing to move deep, we'll do that first. Continuing to move deep, what's going to be the deepest layer in this lateral posterior area of the eye? So retina. Okay, so retina is going to be the deepest. In terms of, we covered humors. So which humor is this here? Aqueous, good. And then behind the lens is going to be the vitreous humor. Function-wise, we've got the iris. That's going to be doing what What does your iris do? There's the muscles. You have a dilator and a constrictor muscle in it. And what is it? Yeah, so constricting and dilating the pupil. Good. You control the amount of light that comes in. We'll look at the retina closer in a few slides. The only other thing we talked about, well, actually no, it's better in the other one. So, zooming in on this region and removing the lens, we switch to now here's cornea. So we're at kind of this junction of cornea to sclera, and so then this dark, super pigmented piece must be the iris. Good. And then, and if lateral to the iris, you have the, so the suspensory ligaments would kind of be in here at working with the lens, but just lateral to the iris, you have a muscle, and it's ciliary muscle here, and then ciliary body, and a posterior to that. So ciliary muscle, which is going to be also part of that movement with iris and the lens, and then ciliary body back here where those ligaments are coming off. Going anteriorly to this region up here, you can see anterior to the corneal scleral junction, you've got this epithelial tissue here. What is this? This is that junction where the inner portion of your eyelid meets up with your sclera, and that's going to be the epithelial lining that rolls over your cornea when you're closing your eye. You think of, when it gets an infection, it becomes an itis, and so when you get some sort of conjunctivitis, and conjunctivitis is inflammation in your conjunctiva. All right, so here's your conjunctiva. Well, part of it, right? The rest of it would have been attached to the eyelid before this segment of slide was removed. Good. And again, that's that mucous membrane that's going to go over your cornea, right? Because you don't want rough skin rolling over your cornea over and over again. That would not be, on one, be very painful, and you'd also scuff up your cornea. And so if you did scuff up your cornea and it kind of had segments that were thinner and thicker, what type of pathology would you expect that we learned about in the sensory lab today and yesterday? Astigmatism, right? So odd thickness and thinness in the cornea, so you don't see quite exactly clearly in those regions because light's getting bent or refracted differently. Good. Right, so then moving in or posteriorly to the inner two tunics of the eye, you can see choroid here. So again, vasculature. There's a vasculature structure right there. Probably some sort of arterial or something, but you don't have to worry about identifying them yet because we haven't done vasculature. That's next semester. What is all of this, though, in this orientation to the right of the choroid?
That's the retina moving out towards the front of the eye.
Good.
The
Yeah,
back
so this is,
the colored part is all the way to the left.
yeah, okay, good. So here's retina, and you said moving to the right is towards the eye, so then internal segment of the eye here, this would be external eye, and just after the cord we expect to see sclera out here. So internal eye on the right, external eye on the left. So in terms of light coming through, where is the light coming from, left or right? The right. So light coming through here and going through all these layers. Good. So starting internally, what's the first thing that light goes through? Here's our ganglion cells. Good. That second layer right here is the ganglion cells. They're nice round cells. They're usually pretty much right in a line, one after the other, just single file. And then deep to them, so a little more internally, is where you're going to have the optic nerve fibers that they're projecting out in towards the inside of the eye. Going deep to the ganglion cells, we have the inner plexiform. Good. And so that's going to be nerve fibers in between these ganglion cells and these guys, which are part of the internuclear layer, and these cells are the bipolar cells. Good. And
the
then
anaprene cells, that's what you call them in lecture.
oh,
The like ones that connect all the way. Yeah, the one that run sideways between ganglion and bipolar cells, and then run sideways up.
that's what the bipolar cells are. Anacrin might be another name for them, but I don't know. So that's more than
Alright,
possible.
hold on, let me get a 10 slot. Will you tell me what they're called again?
Well, so
What you call them?
I'm calling them bipolar cells. If
No, I mean between the ganglion and the bipolar cells, you call it anacrin.
oh, so you mean this layer here instead of calling it inner plexiform? She put it.
Inner plexiform. Okay.
Okay. Yep. Okay. All right. And so this would be your inner nuclear layer, your inner plexiform. These are the bipolar cells because they're running between ganglion cells, and then these cell bodies over here, which belong to the rods and the cones too, both of them, rods and cones mixed in here. And so this is the inner plexiform. This nervous tissue area between the bipolar cells and the rods and cones must be the outer plexiform. Good. And then once we move past this outer nuclear layer where the rods and cone cell bodies are, you then get to this kind of pink hazy layer, which is where if the cell bodies for rods and cones are here, this should then be where the so the retina pigment layer is going to be this dark band. Before you get to the retina pigment layer, you have to have the functional unit of the rods and cones, the actual rod or cone-shaped piece. And then pigment layer, right here. And you can see pigment layer is real dark, and that's, again, light comes in from the right, we said, and innervates rods and cones, but some light is not going to get caught in a rod or cone, and you're going to end up with it getting caught in this pigment layer so that it doesn't bounce back into the inside of the eye and end up hitting another rod and cone some other spot and making it activate, so that you have a continuous sensible way of arranging the image that your eyes are taking in and then sending that to the brain in a way it can decipher.
I have a question.
Yeah.
What did you say was before the pigment layer?
So this retina pigment, this pigment layer here for the retina, so this layer, okay, so that's going to be like the functional units of the rods and cones, the actual rod or cone-shaped part. So if you remember that EM that has a rod and a cone like laying out on it, one is kind of cone-shaped and one is rod-shaped. So yeah, it's like this part where it's actually like a cone or a rod. They're going to be here, and then their cell bodies are going to be this dark outer nuclear layer. All right? Okay. Good. Okay. So then so now this is an EM of rods and cones. You can see the rod is rod shaped and the cone is cone shaped. And that's it for eye. Moving along. Now we're out of the eye and into another sensory organ. What sensory organ? This is not quite a panoramic, but what sensory organ must this be? Okay, so the organ of Corti is here in the middle, and then so if the organ of Corti is in here, it must be what? The cochlea. Good. And then so if we're at the cochlea, in terms of the gross sense organ, it must be the ear. Okay, good. So ear, cochlea, organ of Corti. All right. We didn't look at it in the slide, but what's just opposite of the organ of Corti? You have that other portion of the inner ear. And the organ of Corti is your sound reception, right? And you've got that other thing. It's got the semicircular canals and the utricle and the saccule, and it's picking up your linear and rotational acceleration. Vestibular apparatus. Good. All right. Good. All right. So be aware of that. Also, we didn't look at it in slide, but we looked at kind of blown up gross anatomy structures of the vestibular apparatus. The cochlea is named the cochlea because it's shaped like a what? A conch. Sure, it's a snail shell. Good. So you've got that winding structure, and so this is one slice of that tube that's winding around itself in this conch sort of shape, like a little tower. And so, structure-wise, what were the important regions that we could see at this level? There's more finer structures that we can see if we zoom in, but the way we're looking at it right now, first off, you see these lumens, right? There's 1 lumen, 2 lumen, 3 lumen. In this orientation, the top 2 lumens are divided by this very, very thin, slender, simple squamous kind of membrane thing. So what is this larger membrane more on the left named? It's a scala. So maybe if we talk about the membrane first. So this thin, slender simple squamous membrane is which membrane for the structures of the cochlea? So the vestibular membrane, and the vestibular membrane separates scala vestibuli from the cochlear duct. And then down here we have scala tympani. Okay, and then scala tympani and scala vestibuli, the two scala have perilymph in them. So the whole cochlear duct vestibular apparatus area is all fluid filled. So you have perilymph in here, endolymph in the cochlear duct. If we zoom in where the organ of Corti is located, you can see, so now here's a segment of the cochlear duct, and then down beneath would be scala tympani, right? So all we've done is gone into this region and just zoomed in. So floating up here a little higher would be that simple squamous vestibular membrane separating cochlear duct from scala vestibuli. So if we look just at the organ of Corti, you can see there's some numbered areas here. What were the important structures for the organ of Corti when you look at the organ of Corti in divisible pieces? Got two more membranes, right? There's this membrane up here that kind of looks like a foot, right? You can see the foot and then the leg piece out here. What is that one? The tectorial membrane, right? And then down here at the base, you have the tympanic. Tympanic, remember, is that border between external and middle ear, and that's where your sound waves and kind of strike the tympanic membrane and go to the malleus incus and stapes. So the base for the organ of Corti is the basilar membrane. It's the base of the organ of Corti. And again, tectorial up here at 3. And then these cells kind of look like they've got little wisps coming off of them, or like hair. Okay, good. So they're hair cells. And then we learned about 2 different populations of hair cells. And so there's 1, and then there's 2. Which is which, and what are their names? So we had hair cells, and we learned that there's inner hair cells and outer hair cells. If we zoom back out away from the organ of Corti and look at kind of more of the gross structure, this is just a portion, again, of the cochlea, and there's going to be an outside to it and an inside to the cochlea based on, there's some indicators you can use here to figure out where inside and outside is. So at this level here, does anybody remember the big indicator? If you look at the whole kind of organ of Corti and the area around it, what's going to tell you what's inside and what's outside?
Can you maybe figure out which way it's coiling?
So your big, the big thing is your vestibular membrane. The vestibular membrane is always going to head towards inside. The slope is it's going to slope down towards the inside, unless for some reason it's upside down, in which case it will slope up towards inside. But it's going to head towards inside. So here's your tympanic, or not tympanic, tectorial membrane with the vestibular apparatus, and they're heading towards the inside. If you look out here, you see the spiral ganglion, that nervous tissue. That's an inner cochlear structure. It's not present on the outside area of the cochlea. Everybody all right? Is that a phone or a binder? Okay. All right. So spiral ganglion, the vestibular membrane is heading towards the spiral ganglion. This is going to be the inside. So again, if we zoom in, now we can see the 2 hair cell populations. Again, here's the tectorial membrane, and it's rooted on the left. So this is inner, and here's outer. At this level, the other thing to look at is the tectorial membrane itself. You can see the leg comes down, and then here's the foot part, and the foot points out. The foot points out, it points away from the spiral ganglion, it points away from the center of the cochlea. So again, this must be outer, because the foot is pointing out, the foot's pointing out this way. Here's outer hair cells. Again, these are going to be the inner hair cells. Right. And that is what I have slide-wise. Does anybody have any questions about the slides that we're going over? Okay, all right. So if you're in my sections, this is just a reminder again that with the cranial nerves, putting the cranial nerve number will not be credited. You have to know what its name is. You don't necessarily have to spell it correctly, as long as I can, again, phonetically figure out what it is you've written, you're good, but you do need to know its name, not its number. And just like with the quiz you took last week, you need to have kind of a general idea of its function. And then again, kind of already talked about it with the hormones, except for T3 and T4. I don't want acronyms. You need to know what that acronym stands for. So not LH, but luteinizing hormone. Not MSH, melanocyte stimulating hormone. Alright? And then, I'm not going to go over them verbally with us as a group. We've already spent a lot of time with slides, but just so you know, these are also part of the PowerPoint. When I post this on Alima, you've got limbic system stuff again, cranial nerve stuff again posted with this, and then also a kind of reminder about the Brodmann's area, because that's now kind of more in the past. We looked at the different regions with Brodmann's area, what they did, the lobes, what they did, Wernicke's and Broca's area. So they're at the end of the PowerPoint also. So, alright. And again, does anybody have slide questions? We're all good? Okay, so the stuff that's out, in terms of important things, there's a skull out. Again, just like always, broom straws with skulls. There's a small vial that has the ossicles of the inner or the middle ear out. Same thing again, always stick with the bone. None of the microscopes are focused on anything, so if you're trying to find something or you think it's not on something that's important, find me. I'll be happy to put the microscope where it's supposed to go. Again, reminder with the hands that are out, we have images on both sides of them. One side at the end is the important side, so make sure that's the side you study.